Today we'll be talking about biochemistry. This is the first podcast on biochemistry. We'll be looking at two others as well. Today we'll talk about amines, preparation of amines, amino acids, and zwitterins, or Zwitter ions. The assessment criteria is fairly long for this topic. You need to know the classifications of amines and understand how they act as Bronsted-Lowry bases. You'll have to be able to explain the difference in base strength between ammonia, primary, secondary, and tertiary amines. And you'll have to understand the mechanism of the formation of amines from haloalkanes and the preparation of aromatic amines. You'll find out a little bit about amino acids and identify the fact that they're usually optically active. And we'll also again look at zwitterins or Zwitter ions and understand the conditions in which they take place. Perhaps the best way to look at amines is to look at the ammonia molecule. An ammonia molecule is 3 hydrogen atoms attached to a nitrogen. There are no alkyl or aryl groups attached. Primary amines are an NH2 group attached to an alkyl or an aryl group. Secondary amines is an NH group attached to 2 alkyl or aryl groups. And tertiary amines, you can see where this is going, 3 alkyl or aryl groups. Amines are bases according to the Bronsted-Lowry definition. If a molecule is a proton acceptor, then it's a base. The nitrogen atom forms a dative or covalent bond with many of the molecules and atoms it interacts with. The following video reinforces our understanding of the different theories of acids and bases, specifically the Bronsted-Lowry acid theory.
The Arrhenius theory of acids and bases defines an acid as a substance that releases protons in solution, and a base as a substance that releases hydroxide ions in solution. We know that ammonia can act as a base and exhibits alkaline properties, but contains no hydroxide ions. How is this possible? In this lesson, we will learn about the Bronsted-Lowry theory of acids and bases, which fills in the gaps in the Arrhenius theory. You have learned that acids are substances with a pH lower than 7. We can test for acids using universal indicator solution, or paper, which turns red or orange, all depending on the strength of the acid. Blue litmus paper also turns red in the presence of an acid. An example of an acid that you are likely familiar with is hydrochloric acid. Hydrogen chloride dissolves in water to give hydrochloric acid. Hydrogen chloride will dissociate to give a proton and a chloride ion. The proton is not just released, but it is transferred to another substance. For our example, water accepts the proton to form a hydronium ion. A hydronium ion is also known as an oxonium ion. A Bronsted-Lowry acid is a substance that releases or donates protons in solution, in our case, hydrogen chloride. A Bronsted-Lowry base is a substance that accepts protons in solutions, in our case, water. Notice that the definition of a Bronsted-Lowry acid or base is not a function of pH. So an acid-base reaction, according to the Bronsted-Lowry theory, involves the transfer of a proton from one substance to another. In the introduction, we mentioned that ammonia acts as a base even though it does not release hydroxide ions, which is stipulated in the Arrhenius theory. When ammonia dissolves in water, it accepts a proton from water. Notice that this reaction also involves a proton transfer. Can you figure out which substance is acting as an acid in this example? Which is acting as a base? Please pause the lesson to think about this and resume when you are ready. In this example, water acts as a Bronsted-Lowry acid because it releases a proton. Ammonia acts as a Bronsted-Lowry base because it accepts a proton. So a base, according to the Bronsted-Lowry theory, does not need to have a pH greater than 7, or turn red litmus paper blue, or turn universal indicator solution or paper blue or purple. The only requirement is that it is able to accept protons. Have you noticed that water acted as a base in our first example, but as an acid in the second example? Substances that can act as either an acid or a base are amphoteric. In conclusion, according to the Bronsted-Lowry theory, an acid is a substance that dissociates to release or donate protons. A Bronsted-Lowry base is a substance that accepts protons. Therefore,
we need to be able to explain the difference in base strength between ammonia and the three types of amines. As I said before, we can think of amines as substituted ammonia molecules. And amines act as bases because of the lone pair of electrons on the nitrogen. The nitrogen atom donates its lone pair to a hydrogen ion, therefore it's a base. Tertiary amines are not as basic as secondary amines. And that is true because they only have one hydrogen attached rather than two as a secondary amine would have. And it would have trouble stabilizing itself and ionizing to lose that one hydrogen ion. The strength of amines on bases depends upon the availability of the lone pair. And you can see in this trend that ethylamines are stronger bases than ammonias or phenylamines. And phenylamines are the weakest because of the delocalized electrons that are in the benzene ring that allow the lone pair around the phenyl portion of the benzene ring to move toward the delocalized electrons. The formation of amines from haloalkanes is done by nucleophilic substitution. Now, the ammonia, in order for this to work, must be hot and there must be an excess of ammonia. If not, we have the chance of secondary amines being formed. Nitriles can be formed by a reaction with the cyanide ion. This also needs heat to work. Let's take a look at the phenylamine creation because it is the beginning of the process of the synthesis of dyes, perfumes, and many other aromatic compounds. Because of the lone pair on the nitrogen in the NH2, and because of the delocalized electrons in the benzene ring, the lone pair on the nitrogen are delocalized into the benzene ring. So instead of a nucleophilic substitution, it is more likely to be attacked by an electrophile. And you can see if you add aqueous bromine, you get a triple substituted benzene ring with bromine. Now, The acid and base behavior is unique because if you look at an amino acid, it contains both a carboxylic acid group and an NH2 or an ammonia group. If the R group contains another COOH or carboxylic acid group, then the amino acid demonstrates acidic behavior. If the R group contains another NH2 group, it demonstrates basic behavior. You have, for example, in that condition, two sets of unshared pairs on the two NH2 molecules attached to the amino acid. And therefore, by the Bronsted-Lowry definition, it would demonstrate basic behavior. If there is an alkyl group attached, neither a carboxylic acid nor an ammonia group, then the amino acid is neutral. Now, amino acids are usually optically active. If you looked at that picture, you would have seen a carboxylic acid on one side, an ammonia group on the other side, some sort of R group, and then hydrogen attached to one carbon. Those are four different groups, and the chances of optically active isomers is very high. Optical isomers play a very big part in living cells. We as an organism and other mammals distinguish one optical isomer for another through enzymes. We'll talk about enzymes later on in biochemistry. It's interesting to note that almost all the amino acids found in proteins have the L configuration. Now there are some D amino acids, but they are found in lower life forms, microorganisms, cell walls of bacteria. It seems as if the enzymes that we use to synthesize proteins have evolved to primarily use only the L type of optically active isomer.
Okay, so in the last video we talked about naming enantiomers and how they're different compounds, they're non-superimposable mirror images. But what's really interesting about enantiomers is that they actually have almost all their physical properties are identical. So these are a pair of enantiomers that we've talked about earlier, right? Besides giving them two different names, how would we distinguish between these two compounds, which is very difficult because they have exact same melting point, exact same boiling point, the same polarity. They share most other physical properties. So a good question would be, how do we even know that these two compounds are different or that they're two different compounds? How could we isolate one from the other if they have the same boiling point? We couldn't distill them apart like we did in the lab. What we're going to find is that they share almost all the same physical properties, one of which that's different is the extent to which they rotate plane polarized light, which we're going to call optical activity, and I'll talk to you more about plane polarized light in a second. It was discovered about over 100 years ago by this guy with an awesome beard named Emil Fischer that chiral compounds, enantiomers, can rotate plane polarized light. And he discovered this with a compound that's produced by grapes and wine fermentation called tartaric acid. Notice that tartaric acid possesses two stereocenters. The mirror image of minus tartaric acid is plus tartaric acid. They're not superimposable. What he figured out, actually, looking through a microscope, he could see that there were two different crystal forms that were actually mirror images of one another. So he physically separated out those two crystals, dissolved them in water, and measured the extent to which they rotate plane polarized light. One rotated it in the positive direction, the other in the negative direction. So that was
Now, as we said before, on one side of an amino acid, there's a carboxylic acid portion, And on the other end, there is an ammonia portion. However, each molecule, each amino acid can interact with itself, and it can provide from the carboxylic acid to the base a hydrogen ion. Therefore, we have what is called a zwitterin, or a Zwitter ion. It comes from a German word. This is our first video podcast on biochemistry. So I think it's best to have us start our thinking by looking at ammonia and the Bronsted-Lowry definitions. We'll look forward to see more about amines and amino acids in the following podcast.